This is Module A5, Objective 2, and in this objective we would like students to be able to sketch and label a typical neuron, describe the functions of all of the components or cellular organelles of the neuron, classify neuron on the basis of their structure and function. When we look at all neural tissue, we find that we have two categories of cells. We have the neuron and the neural glial, or glial cells. And this objective will look at neurons, and the next objective will examine all the neural glia found within the body. The structure of a neuron, the typical neuron looks like this, and we've already seen in the lab we can identify the cell body, this region in blue. We note that the cell body has a number of projections extending off of it. The short branched objectives are the dendrites, here in yellow, and there is usually a longer projection known as the axon, here in red. So these are the three major portions of the body of the neuron. We find that neurons contain a very large nucleus and nucleolus. They have a specialized cytoplasm that is known as perikaryon, and they have a lot of mitochondria for energy production. We need to kind of think again, what is the function of the nucleolus? The nucleolus is the site for ribosome production. And what is the function of the ribosome? You should be thinking protein production. There are a number of very darkly staining structures in the neuron that are known as chromatophilic substance. The old name for these were Nissl bodies. Chromatophilic substances are areas that have a tremendous amount of rough endoplasmic reticulum and free ribosomes. And again, when you see that a particular cell has a lot of RER and ribosomes, you should start thinking, why? Why would we expect a cell to have a lot? So this leads you to believe that there is a tremendous amount of protein production. And you should start thinking, well, what type of protein does a neuron produce? And this is something we'll come to in a bit. The neuron also has its own cytoskeleton. We have neurofilaments and neurotubules. These structures replace the microfilaments and microtubules of a normal cell. So they create the scaffolding and help support the shape and structure of the cell. We also find neurofibrils, and these are just bundles of neurofilaments providing support to all of the structures that extend. So when you consider the dendrites and the axon, these are all supported by these neurofibrils. Dendrites are highly branched, and dendrites themselves contain a number of very small processes. Here we have a dendrite, and you can see all of the little projections extending off of it. That should, this suggests and is basically an increase in surface area. And it is the dendrites that receive information from other neurons. The axon itself is very long, and the axon carries the action potential, or the nerve impulse. If we look at the axon itself, it has its own type of cytoplasm known as the axoplasm. And the axoplasm has a number of organelles. It has neurotubules, neurofibrils, and enzymes. And the axoplasm is surrounded by the axolemma. This is the membrane that covers the axon. When you look at the axon itself, we see that there is a very thick section of the cell body that precedes the axon, and this is identified as the axon hillock. And the axon hillock is associated with the initial segment of the axon. The axon terminates in fine extensions that are known as telodendria, and telodendria will terminate in synaptic terminals, and this is something we've seen when discussing the neuromuscular junction. The synaptic terminals are expanded regions at the terminal ends of the axon, and we've already seen that the synaptic terminals are filled with synaptic vesicles. What do these vesicles contain? They contain neurotransmitters. Between the synaptic vesicle, between the synaptic knob, or the terminal region of the axon, we find a synaptic cleft. And this is an area where one neuron communicates with another cell. The second cell can be a neuron, it can be a muscle fiber, it can be a gland cell, but this is where communication happens. And again, the neuron does not directly articulate, it does not directly connect to these other cells, but there's always this synaptic cleft, this space left between. When we look at the interaction of neurons to either other neurons or other cells, we can identify a presynaptic cell and a postsynaptic cell. So we should probably start with the identification of the synaptic cleft. If you remember, your telodendria end in a little enlarged pouch, and this is separated from the other cell type. This other cell type can be another neuron. It can be, again, a muscle or gland cell. And we refer to, in this situation, here we can identify the cleft. And we have a presynaptic cell and we have a postsynaptic cell. Okay, so the way these are organized is associated with the direction of the action potential. The action potential is going to travel down in this direction. And in doing so, we can find a presynaptic cell, a cell that is before the synapse, and a postsynaptic cell, one that is after the synapse. Looking at the synaptic knob, we find that this is an expanded area of the presynaptic neuron, and it contains synaptic vesicles that hold neurotransmitter. What is the neurotransmitter that we discussed in Module A4? This is a neurotransmitter found at all neuromuscular junctions. This is acetylcholine. if I could spell it right. Neurotransmitters are chemical messengers. They're released at the presynaptic membrane, and they have their effect on the postsynaptic membrane. They're broken down by enzymes. If you remember, the acetylcholine esterase is the enzyme that breaks down acetylcholine. And then they're reassembled in the synaptic knob. Neurotransmitters tend to be protein. And this will explain why we have such a dense population of chromatophilic substances, why we have so much rough-ER, why we have so many ribosomes, because we need a lot of these organelles to produce the neurotransmitters that are protein. Types of synapses, we've already seen the neuromuscular junction. The other type that we're going to introduce is the neuroglandular junction. This is a synapse between a neuron and a gland. We've already seen this picture. This is actually a micrograph showing the terminal axon with all of the little vesicles, and you can see them here. When we look at neurons, we find that we have 4 structural classifications, and the first classification is referred to as the anaxonic neuron. Anaxonic neuron. These are very small. They're generally associated in the brain with in close relation to sense organs. And we find that all the processes of the cell look the same. You do not have short dendrites and a very long axon that we would see in a typical neuron, but they all look the same. They're nearly impossible to identify which of these processes are dendrites and which one is the axon. The second structural classification is a bipolar neuron. Again, these particular neurons are small, and they're usually associated with sense organs. Sight, smell, organs of that nature. And in bipolar neurons, we have one dendrite and one axon, hence bipolar. There's 2 polarities. There's a dendrite and there's an axon. Unipolar neurons are found as sensory neurons of the PNS, or the peripheral nervous system, and they have a very long axon. In fact, this entire length is the axon, and you see that you have a fusion. You have dendrites that are fused to the axon at one end of the cell, and you have the cell body that is separated. Let me just kind of increase this picture. Here's your cell body. You then have a process that then goes in either direction, one way towards the terminal axon and the other way towards the dendrites. So this is unipolar. And then lastly, we have the multipolar neuron. This is by far the most common neuronal type. This is the one that we have already seen in lab and earlier in lecture. Multipolar neurons have very long axons and multiple smaller dendrites. Functionally, we can classify a neuron as being sensory, motor, or a class known as interneurons. You should think sensory is associated with the afferent division of the PNS, and motor neurons are the efferent. Afferent heading towards the brain, efferent moving away from the brain, carrying a signal from the brain to the body. And interneurons are just as they seem. They tend to sit in between these other 2 types. Sensory neurons, they monitor the internal environment as well as the external environment. And these neurons tend to be unipolar. The cell bodies tend to be grouped in little enlarged structures known as ganglia. When you look at a nerve, and this is something we'll look at a little bit later in lab, often you have these enlarged pouch-like regions, and this is where the cell bodies of the neurons tend to lie, with their axons running off in either direction. And sensory neurons are going to process. They're processes that extend from the sensory receptors in the body, such as those Merkel cells, all the way up to the central nervous system. Motor neurons will carry instructions from the central nervous system, the brain, and the spinal cord to the peripheral effectors. And this is, of course, via the efferent division, or the efferent fibers. We've already seen that there are two major efferent systems. We have the somatic, you should think skeletal muscles, and we have the autonomic, and with that you should think smooth and cardiac muscle, as well as some others. And then lastly, the interneurons. These are mainly located within the central nervous system, the brain and the spinal cord. And these neurons tend to lie between sensory and motor neurons. Their responsibilities include distribution of those sensory information, and they also play a very pivotal role in coordinating motor activities. In the brain, interneurons play an important part of the formation of memories, learning, as well as the planning regions of the brain.